Meiosis is a process that contributes to genetic variety. It occurs in reproductive cells, producing sperm and egg cells, also known as gametes. The starter diploid cell is primary spermatocyte in males or primary oocyte in females. The goal of meiosis is to go from diploid to haploid cell and make every nuclei unique. Meiosis begins an interphase where cell growth occurs and DNA gets replicated. In prophase 1, the nuclear membrane disappears and spindle fibers form. Chromosomes condense and line up with homologous pairs. Chromosomes form tetrads and do a synapse before leaving each other and exchange DNA. This is called crossover. They are now called recombinant chromosomes, meaning each chromosome has pieces from its other homologous chromosome. They are no longer sister chromatids because they have different alleles. After prophase 1, you have massively increased genetic diversity. In metaphase 1, chromosomes are lined up in pairs in the middle of the cell and are about to finish synapse. In anaphase 1, chromosomes get separated from their homologous pairs and are dragged to opposite ends of the cell. After metaphase and anaphase 1, genetic diversity is increased again. This is because the combination of where chromosomes line up and which ones they end up with is completely random. During telophase 1, 2 new nuclei form. In cytokinesis, 2 new smaller cells are formed, both with their own nuclei, but they are now haploid, which means they have 1 copy of each chromosome. PMAT repeats, and in prophase 2, the nuclear membrane dissolves and spindle fibers form. However, crossing over and genetic recombination doesn't occur again. In metaphase 2, chromosomes line up in the middle of the cell, this time in a single phi line. Spindle fibers attach to the centromeres. In anaphase 2, the sister chromatids of each chromosome separate and are dragged towards opposite poles. After metaphase and anaphase 2, genetic diversity increases again because recombinants line up and separate completely randomly. During telophase 2 of the meiosis cycle, nuclear membranes form around each unique set of chromosomes, and the chromosomes decondense. Cytokinesis splits the chromosome sets into new cells, forming the final product of meiosis, 4 haploid cells in which each chromosome has just 1 chromatid. This results in 4 genetically unique gametes, or reproductive cells. Meiosis is a reduction division because we started with 1 big cell that had 6 homologous chromosomes and ended with 4 smaller cells that each had 3 chromosomes. The final result was 4 new genetically different cells.